Good afternoon, viewers. All the students are welcome to this BISEC series of presentation in microbiology. Today's topic for discussion and presentation is all about enzymes, especially the general characteristics, types of nomenclature systems, and modes of its classification. With me, I have Sri Rikin Bhai Parikh to take part in the discussion. The first thing I would like to tell about enzyme is that the term was coined as late as in 1800 to describe the effervescences observed within the fermented broth by the yeast. The molecules called enzymes were first crystallized by J.B. Sumner in 1926 in terms of urease, and it was demonstrated by him that enzymes are in fact protein. Now, these class of compounds are highly efficient and extraordinarily powerful molecules which are acting like catalysts, and because they are catalysts of the biological nature, they share most of the chemical properties of the typical chemical catalyst. That is to say, they are not consumed during the reaction, they just speed up the reaction rate, etc. The enzymes are in fact occurring in the diverse kind of varieties within every living cell. In fact, a very simplest kind of single cell may contains hundreds of different types of enzymes, and each one of them in numerous copy numbers. Together, this array of enzymes are capable to catalyze number of biochemical reactions which constitute the entire physiology of the organisms. And in fact, it is these metabolic differences and enzymatic reactions which makes the basis for differentiation within the different forms of life. This class of molecules are possessing lot of unique properties which makes them most wonderful molecules, or they can be regarded as the miracle molecules. The enzymatic reactions which are taking place are of a nature that are just not possible under ordinary circumstances, that is at the physiological conditions, unless the reactions are aided up by the enzymes. And these enzymes are in fact able to speed up the reaction to great magnitude, that is up to 10 raised to 3 to 10 raised to 20 times fast than the reaction catalyzed in a non-enzymatic way. Now, important thing about enzymatic reaction is that they proceed towards the attainment of equilibrium status in a very rapid manner. But interestingly, the point of equilibrium is not altered, that is, position of the equilibrium remains the same, whether the reaction is enzyme catalyzed or not. Now, another important thing is that the activation energy required by the molecule to undergo reaction is lowered down, and for this reason, the chemical reaction, enzymatic reaction, can take place at a much lower energy levels without requiring additional energy in the form of heat or in any other forms. These reactions are in fact substrate specific. Each enzyme is very much choosy in selecting its substrate and is able to act upon only and only upon the specific substrate. Now, of course, there are exceptions wherein some enzymes are able to act upon group of compounds, class of compound, but still, despite that, the specificity is maintained. The enzymes are also specific in terms of stereo forms. Enzyme can distinguish between the optical isomers and stereoisomers of different types. The reaction which is taking place in the presence of enzyme are free from different type of byproducts. So in a way, it is like running over the express highway where there are no additional exit or entry points unless there are few specified ones. So when reaction is catalyzed by enzyme, the chances for formation of unwanted byproducts are minimized, and the entire set of substrate or maximum part of it is channelized towards the final products only. So that is the beauty of enzyme reactions. Now, the enzymes are energy efficient in a way that they are able to couple different type of reactions. Some reactions which are endothermic, others which are exothermic, can be coupled together by an enzyme, and the energy released during one reaction is captured and is utilized in the energy requiring reaction which is going on concomitantly. Now, these class of compounds have also evolved with a mechanism for regulation, and these allosteric regulation makes the basis for control of entire metabolic repertoire. So these allosteric activity, allosteric mechanism of an enzyme regulation is because of the specific structure of the enzyme, which allows the binding of substrate as well as effector molecules to its site. Now, how these enzymes are named and classified is an important area. If large number of enzymes are known and the list is being appended every day, every scientist have been using the name in their own way. So this is going to lead to a kind of chaos where there will be a lot of ambiguity in knowing the enzymes. So until the number of enzymes which were known to us was limited, uncontrolled naming patterns were acceptable and would not be having much influence over the scientific research. But when the number of enzymes are growing rapidly, and the names which have been in use are often misleading, and often they do not convey the precise meaning of what kind of chemical reaction is catalyzed by the enzyme. This can be exemplified by the enzymes, enzymatic names like diaphorase or catalase, wherein it is not clarified that what kind of chemical reaction the enzyme is catalyzing here. Similarly, there are different groups of reactions which are in fact catalyzing the similar reaction, and therefore different names may not be needed. So this kind of examples would lead to a kind of ambiguity in knowing the enzyme, in naming the enzymes. Also, some of the established names, that is the enzymatic names which were used currently, were reconceptualized. A good example is that of synthetase. This name is used only in a distinctive cases where ATP is required and is consumed during the reaction. Whereas in the earlier literature, often synthase and synthetase type of names were used interchangeably. And with this sort of understanding, the compilation of enzymatic names in a scientific way is growing on, and the entries which were initially just 712 in the year of 1961, that is initially, is presently having 3,196 entries, as has been updated in the year 1992. Now, ways of naming the enzymes and the responsibility to do so was in fact assigned to a body of experts, and this body is known as International Union of Biochemists and Molecular Biologists. So the work carried out by this committee with regards to nomenclature and classification of enzyme became a landmark for most of the enzymologists and biochemists worldwide, and it is considered to be the most authentic record of enzyme nomenclature. Now, I would request Professor Rikinbhai Parikh to give idea about nomenclature patterns used in enzyme. I would request him to start with the introduction about the IUPAC body, which has worked out the scheme for classifying and naming the enzymes. Rikinbhai, please.
Thank you, sir. And once again, good afternoon to all of you. As sir has discussed and given you the brief idea regarding what the enzymes are and general properties and some basic aspects of enzymes that sir has discussed. Now, after understanding that concepts of enzymes, the structural properties, the physico-chemical natures of enzyme, now we will move further in the aspect of nomenclature and classification of the enzyme. As Yagnik sir has given this brief idea about the international union which has taken this responsibility for giving the perfect exact classification and nomenclature of enzyme. Initially it was assigned to the IUPAC, that is the body, the name of the body is the International Union for Pure and Applied Chemistry. Then Third International Congress of Biochemistry, in 1955 August, it was initially started. Then set of International Commission on Enzyme was been started, formerly the International Union of Biochemistry, that is also known as IUB. That body has started the nomenclature and classification of enzymes to consider the classification and nomenclature of the enzyme and coenzyme, their units of activity and standard methods of assay, together with the symbols used in the description of enzyme kinetics. So this type of the basic aspects they have covered, that how to give name to any unknown type of the enzyme which has been invented by any scientist throughout the world. And as sir has discussed priorly, so many confusions occurs by the name of scientists, the people who had invented that type of the enzymes, and that was the biggest problem. But now with the help of this International Union of Biochemistry, they have suggested this type of the working ideas and principles. They have given two types of the naming procedure. First one is a common name, that is a trivial name, and a systematic name. Systematic name is somewhat what we are interested in, that how this systematic name come arise and which type of the methods and principles are used for giving the systematic name to any trivial type of the enzyme. In this trivial naming, these are the basic principles or aspects which should kept in our mind before giving any trivial name to any type of the enzyme. General, working and current in use, that should be general, working and current for the use. That type of the name should be suggested by the person who has submitted. The person who has submitted will give the name to that type of the enzyme. Common names, that suffix ase should be placed after the substrate. That was the very primitive type of the idea to give name to any type of the enzyme, that they have to consider that the particular enzyme is acted upon by which type of the substrate. The example what we take, we can take the example of urease enzyme. When we say urease, then it is clear in our mind that substrate should be urea, and we have put the suffix ase after urea, so it becomes urease. Then historic type of the enzymes are also there. The names are there, trypsin, amylase. So that type of the enzyme are not following this, but it is a historically proven and accepted type of the names. After the gene, that is RAK, ATPase, after some characteristic. So these are the different aspects, that after the name of gene we have to put it is enzyme, is suffix, and after the characteristic, that is HSP70, that is a heat shock protein, in case of that also we have to put this type of the ATPase. So this systematic naming goes further, that formed per rules show the enzymes and enzyme as exactly as possible. The name, confusing name should be avoided. Uniquely define the reaction to be catalyzed. In systematic type of the naming, it is very important characteristic that unique, they have to be unique in specific in the reaction what it has been catalyzing, combining the substrate plus reaction type. That was the advancement. that you have to combine not only substrate name, you have to put the suffix ase, but substrate name plus the type of reaction which has been catalyzed, that also you have to add in that. Alcohol dehydrogenase. So as the name indicate, we can understand that it will remove H from the alcohol by the process oxidation. So that all we have put in the name of alcohol dehydrogenase. Alcohol is a substrate and dehydrogenation, that is the removal of H. So that is the reaction being catalyzed. So we have combined both of them, name of the substrate and the reaction being catalyzed. So the name of the enzyme we place, that is alcohol dehydrogenase. The lactate NAD oxidoreductase, it is another example by which you can also understand that what it is doing, that lactate is a substrate, that is a lactic acid, and NAD oxidoreductase means oxidoreductase is you can say oxidation of pyrimidine and NAD, that is reducing to NADH. So this type of the naming pattern was there. Now name ending ase used only for the single enzyme, not to the systems. So now this is another aspect, that when only one enzyme is involved, that is known as single enzyme. In that case, you are just putting down ase suffix after the name of the enzyme, but in case of the system, we cannot do same type of the thing. That is the guideline. Oxidation of succinate by oxygen, the succinate dehydrogenase, cytochrome oxidase in several intermediate carriers called succinate oxidase system. So oxidation of succinate, this is the process which is being catalyzed by more than one type of the enzyme. So it is known as an enzyme system. So we cannot say that succinate deoxidase is the name of the enzyme. Instead, that group of enzymes are involved. So we have to place, we have to say that succinate dehydrogenase system is the correct word for that. So disprove the phenomenon naming when no definite chemical reaction, permease, translocase, replicase, this type of the names can also be used when these phenomena are not definite. The chemical nature of the